So yeah, I'm going to present to you the results of my master project on the whole genome sequencing of PRRS. We use a different approach from the gentleman that just went before. So very, very briefly, it's why PRRS? Because it costs over 800 millions in Canada and the US every year. It's very, very harmful, especially during co-infections with other pathogens. And the PRRS genome, so it has a poly ATL, which is very, very useful when we're trying to sequence it. It's 15 kb in length, so it's quite short. It has 10 open reading frame, and we know that now they're not even different types, they're different species of viruses, but the difference between type 1 and type 2 PRRS is about 40% of difference in nucleotide identity. But I'm just going to focus on PRRS type 2 because that's the only one we have in Canada, except one cases, I think, of PRRS type 1. And so in Canada, we have 5 distinct lineage of PRRS type 2, which are mostly, the most prevalent is 1, 5, and 8, 5 and 8, which are vaccine-like lineages. And the current genomic surveillance strategy, so what we're doing right now is we're sequencing the ORF5, which is coding for the GP5 protein. And we use like the sequence to tell producers if the strain that they have is vaccine-like, and also to what other strain it is related, to what other outbreak. And we also use this sequence to try to trace back like the origin in time and in place of this specific strain that they have. And the problem with this is that the mutation rate is exceedingly high for PRRS. And the ORF5 is also very tiny. It's only about 4% of the whole genome. And it's not a good indicator of pathogenicity because we know that many other genes are implied in the pathogenicity of the virus. And one of the very, very big problem is that during co-infection, a lot of viruses are recombinant, which if we're trying to follow epidemiologically, only looking at one gene, we get completely lost. So the problem that we have is that a lot of information regarding the co-infection, the recombination, and the pathogenicity is lost when the sequence of only one gene is obtained by Sanger sequencing. So we hypothesized that doing whole genome sequencing would lead to a better classification of the different PRRS strain, and it would help directly in the fight against this pathogen and the spread of this pathogen. So our first objective was to show that the whole genome sequencing would offer important advantages, but for the producers. Like at the farm level, it would have advantages. And also we wanted to streamline the workflow for whole genome sequencing of PRRS because we wanted to reduce cost and increase availability and financial viability of whole genome sequencing because it is still quite expensive. So my samples, there were 138 samples from tissue, mostly lung, serum and saliva. They had RT-qPCR CQ values ranging between 11.4 and 34.1, so we tested on a very, very wide range to try to test our limits. We were able to obtain whole viral genome from 64% of those samples. All of these samples originate mostly from Quebec farms. We have a few from Western Canada. And they all date, with 2 exceptions, from 2017 to 2019. And as we can probably guess, the sequencing success correlates quite strongly with the viral load and also with the sample type, with fluid samples being usually much better for sequencing of PRRS because there is a lot less host. So our methodology, I'm just going to go through what we do. I'm not going to explain all the results and all the other stuff we tested, but this is what we landed on doing. So we do homogenization with glass beads, which is pretty standard for lung tissue. The RNA extraction is done using its Zimo viral extraction kit. It's a beta-mercaptoethanol. It's what allows us to recover the most amount of viral genome. We do a poly-A-tailed RNA isolation using magnetic beads, magnetic poly-DT beads, usually used for messenger RNA. And the library prep is done using Nextera XT, or more recently Nextera Flex, which allow us to work with only 1 nanogram of RNA. And as a sidebar, there's something else I've been toying with for a tissue sample. It's using polyethylene glycol precipitation and RNase treatment. So on your left, this is the percent viral read of 3 different samples that were treated different ways. And on the right, this is the improvement ratio compared to the control. So in blue, there's no additional treatment. So you see it's a bit under 1% of viral read. Doing a precipitation with polyethylene glycol 8000, so very big polymers of PEG, I was able to get this to almost 2.5. Treatment with RNAs in green and in purple had absolutely no effect. But when I combined PEG and a high concentration of RNase A to be more precise, I was able to get to almost 6%. The improvement ratio compared to control was 8.5 times more viral read for tissue samples. So this is relevant because this is almost gaining 3 CQ value of what we're able to sequence from tissue samples. So I just wanted to share this because I find this cool, but yeah. Now I'm gonna jump right into my sequencing results. So this is the conservation status of the sequence strains. So this is really the conservation status of all the strains that are currently circulating in Quebec. Not all the strains, but a good representation of the strains circulating in Quebec in the last 2 years. When the line is blue is because this specific spot is completely conserved, and as the blue line drops is because there are more and more variability at that point. The first thing we noticed is we have 3 deletions in many of the lineage 1 strains in NSP2 and NSP7. We don't know what they mean yet. We haven't done any tests on it, but we keep finding them in most of the more recent lineage 1 strains. So this is interesting. And also what is also interesting is that the first quarter of the genome is by far the most variable of the genome. So NSP1, NSP2, and NSP3 are really, really quite variable compared to like even ORF5. We were a little bit surprised to see just how much it was. We had up to 21% interstrain differences in the strain that we sequence. So now I'm going to talk about co-infections because we found 4 evidence of co-infections. This is the phylogenetic tree of the sequence. I'm sorry if the numbers of the strains are a bit too small. But these two strains were found in the same sample, and they share 88% identity. These 2 strains were also found in the same sample. They share 92.36% identity. These 2 strains, they have something that's quite similar that I enjoy, is that one of them, they cluster closely, but one of them is on a very long branch that's kind of apart from anything. This, I believe, is because it's actually a recombinant, and we're going to see that they're recombinant from before, in the next slide, but they're a recombinant that occurred a while back, and they evolved in their own way since. That's why they're alone on the bar, but still related to the other strain. And then these 2 also were a co-infection, but this one is a recent recombinant. If you look at the conservation status between the 2 strains, after the first half of the genome, the strain is completely identical to the others. So probably there was a co-infection. They made a recombinant. One of the parent strain was eliminated from the animals, and what we sequenced was the other parental strain and the recombinant strain. And then the 4th co-infections, the strain are nothing alike. They share only 81% identity. They cluster completely differently. And now I'm going to compare the whole genome sequencing phylogenetic tree and the ORF5 phylogenetic tree. So to indicate, these are the 3 big commercially available vaccines in Canada that are used prominently. It's lineage 5 and 8. And the first strain I want to show is this one. So comparing on if we use only the ORF5 or the whole genome sequence, they cluster completely differently. So this strain is probably a recombinant too between a vaccine-like strain and what we call a wild-type strain. These two strains are the two that I mentioned on the previous slide. They don't look to cluster that far apart, but it's very, very interesting because if you look, these still cluster with the lineage 5 and 8 strains, whereas on the ORF5 tree, they cluster with the lineage 1. So the classification changes completely too. And then we had three other strains who cluster like differently on the tree, but they still stay into the same lineage. This is still relevant because when we're trying to trace outbreak, or I'm trying to say, like, for instance, this strain here was highly pathogenic, so it could be important to know if a strain is really similar or not. So in summary, we have some deletions in the ORF1 that appears to be marker of the non-vaccine-like strains that we have in Quebec. We have at least 4 co-infection in 88 samples in which we were successful for sequencing. We had at least 6 of 92 strains that clustered differently based on whether we're looking at the whole genome or the ORF5 only. So this suggests that whole genome sequencing could offer a better classification of clinical cases in at least 11% of PRRS type 2 cases. I say at least because for the co-infections, I only looked at those that I found 2 complete strains. So since my success for sequencing was about 64%, then I probably missed some co-infections too. So the number of co-infection is probably higher. So the next step, we want to keep sequencing more strains, more diverse strains to add weight to our analysis and to build a tree. And we'd like to reconciliate the full-length genome with the existing PRRS phylogeny models that are based on ORF5. And we also want to start gathering information about clinical manifestation of the various strain to try to identify genetic marker of pathogenicity, and also do that in the non-coding sequence. So thank you, and I just want to thank all our financial partners and the PRRS Symposium to allow me to speak.
Questions for Chris? So when you said you have 4 out of 88 samples you identified as the co-infections, so you know when you analyze those sequences, always there's some quasi species. So how did you define that this is the true versus strain rather than just the quasi species of one strain?
Well, with the quasi species, we're expecting a virus that's going to mutate and differentiate in the same host or in the same herd. So we're expecting a small number of differences. The cutoff that I decided to be was 4%, so more than 96% difference I decided was a co-infection. It's a bit arbitrary, but I said maybe I have confidence in 99% of my sequence. If I'm wrong and they're a bit less, if I have a bit more error, it could go up to 2. So I said 2 both ways would give 4. So more than 4%, it's not mistake, it's not any trouble, it's clearly different strains.